So understanding the neural connections between the brain and behavior is at the very heart of this evening's program, entitled Hardwired for Song and Dance, the Neurobiology of Language and Movement. I now have the pleasure of introducing tonight's speakers, Professor Erich Jarvis, head of Rockefeller's Laboratory of Neurogenetics and Language, and Dr. Constantina Theofanopoulou, an associate research professor at Hunter College. Tonight, they will share with us their fascinating research on the neuroscience of language, movement, and music and movement from a perspective that is rare, if not unique. Erich and Constantina are not only wonderful scientists, they are both trained dancers. And this gives them a unique perspective on these problems, and they are exploring how humans came to be a species that dances, sings, and uses complex spoken language to express emotions, build connections, and share information and ideas. Erich Jarvis is representative of the brilliant and innovative faculty working at Rockefeller. He was born and raised here in New York City and discovered his passion for dance early in life, attending the renowned High School of Performing Arts here in Manhattan. Erich was invited to audition for the prestigious Alvin Ailey Dance Company, but ultimately chose to pursue a degree in science and mathematics at Hunter College. Following graduation from Hunter, he came to the Rockefeller University as a doctoral student in the laboratory of Fernando Nottebohm. Erich received his Ph.D. from Rockefeller in 1995 and continued here as a postdoc before being recruited to the faculty at Duke University in 1998. Erich was appointed an investigator of the Howard Hughes Medical Institute in 2008 and returned to Rockefeller as a professor and HHMI investigator in 2016. Erich is also one of the principal scientists in the Price Family Center for the Social Brain that I mentioned previously. The main focus of the Jarvis Laboratory is vocal learning, the ability to imitate sounds, which is the foundation of human spoken language. This trait is rare in the animal kingdom, present only in humans, songbirds, and just a few other animals. Through studies of animal models, Erich and his colleagues have made highly significant discoveries about genes and neural circuits that play critical roles in vocal learning. As you'll hear tonight, some of these genes and circuits are associated with the control of muscles that enable speech and movement, providing intriguing clues to how language and dance emerged as powerful modes of human communication. Erich's quest to understand vocal learning has led to ambitious efforts to sequence the complete genomes of birds and other vertebrate species. The comparison of these genomes provides unique opportunities to identify commonalities among those few species that have acquired the ability to engage in vocal learning. And he has already discovered a number of genes and pathways that contribute to this trait, and in addition have provided insight into the origins of a number of speech disorders. Erich has received many recognitions for his work, including the 2002 Alan Waterman Award, the highest honor given to a young investigator by the National Science Foundation, and a National Institutes of Health Director's Transformative Research Award in 2019. Our second speaker this evening is Erich's colleague, Dr. Constantina Theofanopoulou, who received her Ph.D. from the University of Barcelona and conducted research at Duke University. In 2019, she joined the Jarvis Lab at Rockefeller as a postdoctoral researcher. Constantina was named a Fellow of the Center for Ballet and the Arts at New York University in 2021. And last year, she was appointed an Associate Research Professor at Hunter College, and she continues her affiliation with the Jarvis Lab. As we all know, language and dance are both integral to social communication and cohesion. Constantina's research has underscored this theme through multiple initiatives, including a specific focus on the role of oxytocin, the hormone associated with interpersonal bonding, in vocal learning in songbirds and humans. She has also developed strategies to use brain imaging and other technologies to understand the neural mechanisms that enable learned speech and movement. Constantina's research in neuroscience has led her to studies in genomics, both independently and collaboratively with Erich. Her investigations also have implications for understanding and treating movement disorders and language deficits. Constantina has received numerous recognitions, including selection as one of Forbes magazine's 30 Under 30 in Science and Healthcare in Europe for 2021, and designation as a next-generation leader by the Allen Brain Institute in 2022. Erich will be the first to speak this evening, followed by Constantina, and there will be time to ask questions of both of our speakers after both of them have concluded their presentations. Now, please join me in welcoming Erich to the stage.
Here we go. Well, wow, thank you for that introduction. And thank you for coming to this event. This is the first time I would say that, or maybe not exactly the first time, but really where we're trying to merge the two topics together about neurobiology of language and dance, or vocal learning more specifically. And in order to study these kinds of traits, we have to do a comparative analysis, like of the kinds of species you see up here, including Jennifer Lopez, the singer. All right, where the species that have this and those that don't. You just can't study humans and mice only. And so, you have to take a useful perspective. And this, like Rick said, you know, I take a perspective from personal experience where, yes, I started out not as a scientist, as a dancer. This is me near the end of my high school years, where I was dancing with the Westchester Ballet Company in a Nutcracker for one of the holidays. And they made me the Arabian guy, maybe not surprising. And I was really, you know, pushing hard to be a professional dancer. But something clicked around that time with my mother always saying, do something that has a positive impact on society. And I did like science as well. And so I transitioned to science and went to Hunter, as you heard, and felt I can, as a scientist, I can do something. But some people ask me, how did you make that switch? And what I have to say, what I learned along the way is that being trained as a dancer and an artist in general and trains you to become a scientist, all right? Both require discipline, hard work, creativity, lots of failure before success, and so on. So I wanted to put that little tidbit in. And what did I choose when I got into neurosciences here at Rockefeller? Study vocal learning as a model for human speech. But when we think about what human speech is, many of us don't realize that it actually consists of multiple components here. And these multiple components are, you know, together make up spoken language, but each one of them by themselves, people don't realize some of those can be found in other species and some others not. For example, vocal usage learning, to use sounds in novel contexts, even though they're innate. And that's pretty common amongst the animal kingdom. But the ability to imitate sounds, vocal production learning, is rare. And so when we put all this together, I define spoken language as a specialized form of learned vocal communication, all right, with some components that is, unique or highly specialized, and other components that are found in many different species. So who are they? The vocal production learners are 5 groups of mammals, including us humans, the cetaceans, those are dolls and whales, bats, elephants, and seals. And there are 5 groups of birds: songbirds, hummingbirds, and parrots. Many people know parrots imitate vocalizations, but they don't realize even a hummingbird can. And so all of these species have close relatives, like chimpanzees to humans or falcons to parrots, that don't imitate sounds. So it's thought that this trait in the 5 mammalian groups and the 3 bird groups evolved independently of a common ancestor. What about some of those other components? Auditory learning. Auditory learning is more ubiquitous. An example is your pet animals. You can teach a dog to understand the sound sit, or siente say, osuwari. Does anybody know what language osuwari is? Japanese. Japanese, right. So people don't, the dog will know that who grows up in Japan, right? But see, you don't, right? That's an example of auditory learning, all right? And a dog can even understand human sentences like get the ball, roll over, and so forth, or you want a pet. But the dog can't say, okay, you got it, I will sit, all right? Instead, a dog goes woof, mostly woof, And it can learn how to, like I said, vocal usage learning, learn how to woof for attention, woof for food, woof for lots of different things. But it won't say, I want food, all right? So, but not all vocal learners are equal. Amongst the non-human vocal learners, the parrots are the most advanced. This is a budgerigar, a small parrot that you see in a pet shop, parakeets, that's been raised with humans for 4 years and learned about 400 words. And Disco could do this. Here's an example.
Never shake a baby bird. That would surely be absurd.
Never shake a baby bird. That would surely be absurd. Are you not a crook? My name is Disco. Cat, meow, meow. Let me guess. Any kid has cheeseburger? Are you a little stormtrooper? Disco, I used to be a spy. And a parakeet. Shoo, I'm a doctor. Not
Can't
a
decide.
I did. I'm a doctor.
So this is quite amazing compared to what that dog does, right? Look, although the bird does get the category right sometimes, like sees the dog and goes meow. There's a cat in that household, right? But kind of gets the exact species, not always correct, but that's still amazing. And Disco can actually recombine words into new sentences. A lot of times they don't sound meaningful to the listening humans, but other times, like you see here, they do. So that's quite remarkable. What about our closest relatives? Here's an example. And when this got published, Coco, a gorilla raised for humans for 39 years, I got calls from reporters and others to ask me, what do I think? Am I excited? And here's the best that a non-human primate closest to us can do.
How about when you're coughing? That was good. You did a sneeze and then a cough. Excellent.
So this is good, it's excellent, but it's not what Disco does, right? Coco's not saying, I'm a gorilla or I'm a doctor, right? What's going on here is Coco has very good auditory learning, comprehension, can understand auditorily about several thousand human words, can even sign many of them, but can't actually produce the learned vocalizations that make up those words. The best Coco can do is voluntarily blow air through the vocal tract without a lot of modulation of the larynx. And so that has, from my career, folks like Fernando Nottebohm and others, has been bugging us for many years to ask this question, why can't some of our closest relatives removed 60 million years from us, can't even say something as simple as apple, all right? Whereas those relatives like 300 million years from us, like these parrots, can go golden delicious, Macintosh, all kinds of varieties and so forth, all right? This has been bugging me for over 20 years. And so some of that answer starts actually when I was a postdoc here, where we found that when songbirds produce their learned song, like these canaries here, just singing, it's a learned vocalization, like you saw the parrot do. That act of singing is associated with a robust increase of what we call immediate early genes, genes that are responsive to neural firing in the brain. This is a brain slice. Here's the front of the brain. Here's the back of the brain. And what's lighting up here is what we call the area exon nucleus in the basal ganglia and the HVC son nucleus in the cortical regions. By the way, Fernando had originally named these structures. And we found that for every song about the bird sang in a half-hour period, you had this one-fold increase, a very rapid gene product. This is the messenger RNA. Expressed in these neurons that are controlling the ability to produce the learned vocalizations and imitate them. And we found that it was a motor-driven response in the brain. That is, the birds had to move their larynx, or try to move it. It didn't require hearing, it didn't require vision, whether or not another bird was around or somatosensory feedback from touch, it had to move the vocal organs to get this response. And what we did is we used this molecular mapping tool to try to identify vocal communication circuits across many different species. And what we found with a colleague of mine, Claudia Mello, who was also once a student here, was that we found in all bird species, colored in blue here, this auditory pathway in the forebrain. Whether you're a non-vocal learner or a vocal learner, supported that all species have auditory learning, even chickens. But only the vocal learners had what I colored in red here, in yellow, pathways that lit up when they produced their vocalizations in the forebrain. And we knew from work in songbirds that this pathway makes up the brain regions that control imitation and in yellow makes up the regions that control the production of the imitated sounds. Exactly 7 in each one of these vocal learning bird groups. And some people said, well, this indicates there must be a common ancestor. And that was bugging me also for 10 years. And I got involved in a large genome consortium, and I was getting bored with waiting for it, so I helped move things along and ended up leading a phylogenetic study here to ask the question, what is the family tree of birds really like? And we did rearrange many relationships, but the vocal learners here are still far apart, especially the hummingbirds relative to the songbirds and parrots. So to argue a common origin for these 7 brain regions, you would have to argue around the time of the mass extinction of dinosaurs, there was a common ancestor of all modern birds that had vocal learning ability, and it was lost at 10 independent times. Highly unlikely. So we think it is independently evolved. What about humans? Did humans and songbirds have a common ancestor that imitated sounds and chimpanzees didn't? I doubt it. So the first lesson we learned in comparing songbirds to humans is that the brain size, this is a zebrafinch brain to scale with a human brain. You can fit 3,000 of these into one human brain. Brain size doesn't matter. Neither does cortical surface folding matter. I argue it's the presence or absence of a particular neural network. And to really ask the question about these neural networks of what differs in, it's similar between humans and songbirds, what we did is we profiled the gene expression patterns, one of those genes that I just showed you and many others, using these genomes of the songbirds, non-vocal learning birds, of human brains, postmortem human brains, non-human primates, and so on. And what we found with these gene expression patterns is that there were, as of to date, with work in the past and work since I've joined Rockefeller as a professor, we have about 350 genes in different brain regions shown here. That blue means downregulated in the vocal circuits compared to the surrounding motor areas. Red means upregulated. And what we find, like for HVC and RA, there is convergent up or down regulation of these genes compared to the surrounding non-vocal areas. For HVC, that matches a particular cell type in the human motor cortex. For the RA song nucleus that projects out and controls the vocal behavior, also matches this region that projects out and controls the larynx. And a part of that basal ganglia again, with that area X, that matched part of the human, what we call striatum, involved in speech learning. It was shown after this, it was involved in speech learning. And when we look at the pattern of these genes, the white signal again is the gene product. We can verify the down or up regulation of many of these genes that we saw using other means, only in the vocal learning birds, not the non-vocal learning birds. And we see also here in the human, there are 2 areas of down regulation of this particular gene, which we call dorsal and ventral laryngeal motor cortex. It's in the primary motor cortex that controls movement, where we're seeing these specializations. And other work from Eddie Chang's group, who he talked here a few months ago, found that these areas where we're seeing the specialized gene expression are the areas that light up the most in his patients when he's trying to remove tumors or epileptic regions when they speak. They're trying to spare the speech areas. So they're exactly those coordinates showing a functional correlation here. So putting all this together, we argued that humans also have evolved similar brain pathways as we see in these vocal learning birds. What's interesting here is that all mammalian species, I argue, we can find a Wernicke's-like area involved in the auditory comprehension of complex sounds, including human speech. This is why dogs can understand sit, or Coco can understand, well, how about coughing? Because I argue they have the ability to understand complex sounds in meaning, including from human speech. So how could this come about? A bunch of those pictures I showed you, when we published them, some groups got a hold of them and said it's intelligent design, because you can't get that multiple times like that. We didn't really have an answer, an alternative at least, until this discovery, where we found that not only the brain regions involved in producing learned sounds light up when they're singing, but when they're moving their bodies, hopping in a rotating wheel, we find neurons directly surrounding all of those vocal learning brain regions light up too, for body movement. And this was the case for all the vocal learning birds. The non-vocal learning species had these movement areas, but without the holes inside of them, meaning that they didn't have the song nuclei. And when we looked at the connectivity of the circuit for the song system compared to the connectivity of the surrounding motor system, it was quite similar with some small differences, important, but differences, but still quite similar in terms of connectivity in their projections now to limb muscles, feet muscles or wing muscles, right, in the case of the bird, as opposed to vocal muscles. And it turns out, a colleague of mine, Steve Brown, at the time, was doing some work with dancers and using PET and other imaging, was able to image dancers doing complex choreographed sequences. And the more difficult he made it, see, the highest activation in the human brain regions are directly adjacent to what we call ventral laryngeal cortex or dorsal laryngeal cortex here, involved in speech production, in Broca's area. And so also at that time, it was discovered that only vocal learners can learn how to dance. All right? And here's an example of a cockatoo, a parrot, that learned how to dance from observing teenagers and listening to music.
Hi, Snowball. So, Irina, this is so much better than Dancing with the Stars. Tell us very briefly what Snowball can do, and then we'll witness it for ourselves.
Well, Snowball can dance to a beat, and he actually adjusts his dancing to fit the tempo of music.
So he gets stimulated visually, but he can do this also auditorily only, just listening to the music. And what I want you to pay attention to is his ability to dance to the downbeat. He's
does he like for everybody to join in?
There's the downbeat. And here's some dancers, several that went to Rockefeller. And who's doing better, the bird or the dancer? All right? So I encourage you to go online and listen to that. But before this discovery, it was thought that only humans can learn how to synchronize body movements to sound. All right? Well, it turns out these parrots do too. Then it was discovered that some songbirds do and elephants do as well. And so putting all this together, we came up with what we call the motor theory of vocal learning origin, that all vertebrate species have these innate brainstem regions here involved in the chicken crowing, the dog barking, and so forth. And they all have this motor forebrain circuit. The white arrows are for learning how to produce movement. The black arrows are for controlling the body movements directly. And I argued that this brain pathway for movement control is replicated multiple times in the vertebrate brain that get connected to different body parts. But in us vocal learners, it got replicated one more time through some kind of genetic change. That new pathway took over the brainstem pathway for innate vocalizations to get this emergent vocal learning pathway that's similar to the surround. Another name we call this is brain evolution by brain pathway duplication. The whole brain pathway duplicates. And so in parrots and songbirds, I said we had two pathways. And what I argue goes on here, in the light green is the ancient parrot pathway just for regular movement control. And the first duplication occurs here with the red dots in the forebrain, 7 brain regions. And then you get a second duplication here in yellow that leads to 2 parallel circuits that I think we also have in the human brain, giving humans and parrots even more advanced abilities to imitate sounds. And I argued that this new vocal learning circuit for tight auditory motor integration then influences the surrounding brain areas to get tight auditory motor integration, not just for the voice, but for the rest of the body to get dance. And so is there any evidence that that could be the case? Could these genes have anything to do with dancing that we're seeing in these songbirds? A former graduate student from Fernando's lab, Nicole Crianza, her group published a few years ago a study where they took studies where 23andMe people, who many of you I'm sure, who send their samples in, those who do the surveys, and they asked people the question, can you clap to a beat, right? That is, can you listen to music and clap to the beat like that? Or do you have difficulty doing that? And there are people that do have that difficulty, and the ones that say they do have genetic differences in a bunch of regions here and these different chromosomes here makes it difficult for those people to maintain a beat in clapping. And sometimes they say they can't dance. But humans can dance better than primates. I guarantee that. I don't care how bad you clap. All right? And then what Nicole's lab did is that they then compared those genes to the genes we found regulated by singing or specialized in these circuits. And guess what? They actually found an overlap between these gene sets, the difficult beat clappers, and the singing regulated genes. And it's not just all song nuclei, but it's particularly the area X song nuclei had a significant enrichment of overlap of these genes associated with inability or difficulty keeping a rhythm. And particularly, there was the biggest association in area X for those genes that directly correlated with the amount of singing that the birds did, like I showed you in that first slide. So this is quite remarkable. What about those genes that are specialized but not regulated by singing, but are specialized even just from baseline, from the time you're born? Like this gene here, downregulated in the song-learning nuclei. Some of those genes, when mutated, is associated with speech deficits in humans, like this child here, Bella. Here's an example of what she does. She's trying to say bubble.
Bubble.
And you can see her coordination is decent when she puts the bubble blower into the bottle. So Bella has a frame shift mutation that causes this protein, this transcription factor that regulates other genes in the brain. It's specialized in the song-learning nuclei, specialized in the speech areas, and it's not functioning properly. And what's amazing here is, like what we see in non-human primates and so forth and others, Bella has very good auditory comprehension compared to the deficits that she has for production of speech. And so it seems as this gene and some others like FOXP2, another transcription factor, when mutated and expressed in a specialized way in these speech areas, seems to affect speech more than other circuits. We don't know yet how Bella dances, but that would be something to test. By the way, her family is very interested in what we learn next from studying these genes. And so how could I explain all this together to wrap up here? Well, I told you about the motor circuits here in green and the new vocal learning circuits in red and yellow that are controlling the ability to produce and imitate those sounds. But where is that auditory signal coming in? Well, of course, it's coming in from the ears. It goes up to the forebrain regions here, the auditory pathway, and then sends that auditory signal into the vocal system. And what I think is going on, my prediction is here that we'd like to study for the future, is that in the vocal learners, you have a greater, tighter integration of auditory motor inputs, and that tighter integration then, let's say, contaminated the rest of the brain, right? And that contamination is a good thing, because it now allows you to synchronize your body movements to the beat of sound in dance. So just to put this in words, is that, if we can prove this, this would explain why the vocal learning species learn how to dance and the others don't. And could we find even genetic differences in the ability to dance according to these genes? So those are some of the questions we'd like to ask for the future. For a human brain, it would look like this, where you have the auditory signals coming from Wernicke's area going into Broca's, but also the surrounding areas influencing them in a way as if it were like a speech pathway, but it's not. It's an arm pathway, it's a hand pathway, and so forth. So coming back full circle, I'm actually starting to study dance now in our lab, right? And I never thought I would do that. And I'm doing it because it's associated with vocal learning and spoken language, but it's also associated with a passion of mine. And apparently I'm attracting people to the lab who are also dancers, like Constantina, you hear from a minute, and Sadie Paez, who's also in the audience here. And these are their profile pictures, but they also have dance pictures like I do. Constantina, her favorite is flamenco. Sadie, her favorite is salsa, and actually me dancing with her on performance right before the pandemic. And so I'm going to pass it on now to the next generation to hear what we're going to talk about for future research. Constantina?
Thank you, Erich. It's really such a great honor to be here tonight, and it is even greater that Erich was able to set up the background of what I'm going to talk about tonight so beautifully. Thank you, Erich. So tonight I'm going to give you an overview of how we're currently addressing the question of what could be linking speech and dance in humans. Because we're currently at this great point that we can finally utilize this knowledge that we acquired from songbirds and parrots and translate it back to humans. And I'm going to do so by first talking about behavior. What is linking speech and dance in behavior? To then get to the brain pathways, the brain regions that fire up while we're singing and while we're dancing. To then get to our current efforts to identify which are the genes that enable us to speak and to dance. I will lastly talk about a specific therapeutic practice that we're currently testing in human populations that show both deficits in speech, but also general body movement deficits, as for example, those that we see in dancing behaviors. For example, one population that we're testing is the Parkinson's disease population. So let's start from behavior, because as Erich said, and as someone could read in the literature, what is really common between speech and dance is that they are both characterized by what we call a tight auditory to motor integration. So let's delve into this. Let's start from speech, because oftentimes it is not so easy to appreciate that speech is a motor behavior, because we cannot see our laryngeal muscles moving while we're speaking. But definitely, as I'm speaking to you right now, my laryngeal muscles are in constant motion, and they're actually accompanied by movements that you can't see in my orofacial musculature as I'm moving now my lips, my tongue, my jaw, right? And this is something that you can perceive even better in this magnetic resonance imaging of this person while they're producing just some minimal sound.
E, E-P, E-T.
And if we really want to get a close-up to what's happening in the laryngeal muscles, you can actually see here my own laryngoscopy. So this is my own larynx, as I'm trying to produce this prolonged vocal sound. So this was really fast, right? The vocal folds are really moving non-stop. And I think that after having watched these videos, it really goes without saying that speech is also a motor behavior that, of course, relies on this tight auditory to motor integration, which is something that we can understand because, obviously, in a dialogue, we have to first auditorily perceive what our interlocutor has to say in order to afterwards initiate our response and hence initiate our laryngeal muscle movements. But what we tend to appreciate less is that we also need to constantly perceive our own voice's auditory feedback. We need to hear what we're saying constantly in order to be able to successfully speak. Great experiments in humans have shown that if we mask our own voice's auditory feedback, our voice, our speech really breaks down in several measurements. Our pitch, our articulation, our intensity, everything changes in our speech. So speech really relies on this auditory to motor feedback. What about dance? Well, dance is more visibly a motor behavior, and of course, it really requires this auditory to motor integration because it is even, I guess, counterintuitive to look at these videos without imagining that these dancers' body movements entrain to a beat of the music that they're listening to. And of course, dance is a motor behavior that it requires the activation of several body muscles at a time. And here you can see, for example, some of the body muscles that get activated while a dancer that looks strikingly like me is trying to pull off this balletic arabesque. So what is truly exciting about speech and dance is that these two behaviors are not only linked evolutionarily, as Dr. Jarvis said, they're also linked developmentally in human children development in particular. What is happening in particular is that there is an overlap in the timing that human children fully acquire their ability to perceive the musical beat and to entrain to rhythm, and when they fully acquire their ability to produce the full repertoire of vocalizations in their mother languages. So speech and dance are two behaviors that go hand in hand also in development. So let's get to the interesting stuff. Let's get to the brain pathways. What is happening in our brains while we're speaking and while we're dancing? So first for speech, thankfully there is a wealth of evidence showing what's happening in the brain while we're speaking. And together with Dr. Jarvis, we have made this important, I would say, effort to put together in one paper all these brain regions that have shown consistent activation while we're speaking. And you can see them here in blue, while in yellow, you can see some brain regions that get activated just some milliseconds before we start speaking. And we're saying that this is a pathway that is involved in what we call speech preparation. Now for dance, there aren't actually that many studies that have tried to identify what's happening in our brain while we're dancing. But definitely from the studies that are out there already published, there is an overlap between the brain regions that fire up while we dance and while we speak, that you can see here in green, although of course there are several regions that you can see here in pink that get more activated in dance-related tasks rather than in speech-relevant tasks. But there is a big caveat here. This overlapping coloring here really doesn't tell us a lot because we cannot know whether there is any minor point, any neuronal population in these wide brain regions that get activated both while we're speaking and while we're dancing. Let me showcase this point even further by just focusing on this region of the brain that is called the primary motor cortex that, as Erich said, is involved in the control of several body movements. And I will focus even further in this laryngeal motor cortex that, as its name says, it is involved in the control of the movements of our laryngeal muscles. And I guess you will agree with me if I say that, well, we expect from this region, the laryngeal motor cortex, to be more involved in speech because we have to use our laryngeal muscles in order to speak rather than in dance because I guess we're not using our laryngeal muscles while we're speaking. So if we zoom into this specific area, which is the dorsal laryngeal motor cortex, the upper part of the laryngeal motor cortex, and we want to see from the evidence out in the literature what's happening in this specific region while we're speaking, which are the specific points that fire up while we're speaking, which are here that you can see them in blue, and which are the ones in the surrounding areas that fire up while we're dancing. And you can see them here in pink. And of course, I guess you can see that there is this concentration of these pink points up here and the concentration of these blue speech points down there. But definitely there is this intersection, this overlapping region that seems to be activated in both speech and dance. Again, we should be very careful. This is just my reanalysis of several studies in the literature, and what we need to do and what we're planning to do is to ask this question by studying the same individuals, namely have the same individuals and first have them speak and afterwards have them dance and see what's happening in their brains. Only in this way will we be able to disentangle the question of what is the relationship of the speech and the dance brain pathways in humans. Is it, as in the case of songbirds, as Erich showed, that there are these speech brain regions in humans, like this dorsal laryngeal motor cortex that I'm showing here just as an example, and the dance areas are just sitting adjacent on top of them. They're really sticking right next to each other, but they're not the same brain area. Or is it that in humans, there is actually this in-between green overlapping area that is doing both speech and dance? So we're currently addressing this question through two different ways. First, via electroencephalography, and secondly, via functional magnetic resonance imaging. And I guess it should be easy for you to imagine how can someone speak or dance with an electroencephalography cap on, but it should be definitely difficult to imagine how can someone dance while having to just lie down like this with their head fixed in a scanner. So for these reasons, since we're also dancers, we are creating specific choreographies that are danceable in a scanner and that employ specific arm or leg movements, cyclical rhythmic wrist or ankle movements that can be done in a scanner. And we're also using this inclined board here that permits for dancing leg movements. And very recently, we took some important steps in further developing and optimizing the technology that will render what I call here mobile electroencephalography possible. Because what's happening in science is that you start out with the burning question, right? What's happening in our brains while we're dancing and what's happening in our brains while we're speaking? To then realize that you first have to come up with the right technology to address the question. So this is what we did here. So in this study that I led and coordinated with several other collaborators, such as Sadie Pice here and other collaborators from the University of Houston, is that what we wanted to do was to record brain activity from 5 professional dancers while they were dancing simultaneously on stage, which, by the way, was the first time that it was ever achieved in scientific history for 5 dancers. And so what we wanted to do, the first thing that we managed to do was that we had these dancers dance wirelessly so that they wouldn't be afraid of just tripping with any cables on stage. So we had their electroencephalography caps send wirelessly the brain signals to our computers, which was a very big update in the technology. And we also managed to deal with something that is very tricky in electroencephalography. This is called head and neck movements, because while we're dancing, we need to move our head and neck. But this is really tricky for electroencephalography because it feeds noise and artifacts into our brain data. So by tracking and recording consistently all the time head and neck movements, we managed afterwards to remove them from our data and to have crystal clear and clean data to right now analyze. And here you can see how greatly our dancers were able to move on stage at the University of Houston with the Wi-Fi processing units here in shock-absorbing material that we custom-made for this experiment. And another very exciting part of this project that I want to bring up is that we managed to use their signals from their brain in real time while they were dancing on stage and project it on a screen behind them, which was something that was showing these brains synchrony, these dancers' brain synchrony while they were dancing. So here you can see these dancers' 5 brains that correspond to the dancers and lines connecting them. And the thicker the lines were becoming, the higher their interbrain synchrony. So although we're still analyzing the data, I think that this already suggests that while we're dancing, not only our body movements are synchronized, but also our brain waves, our brain oscillations, the way our brain works. So let's get now the level of the genes and see how we can use this information of finding the exact locations that fire up during dancing, during speaking, or during both of these behaviors to understand the genetic architecture of speech and dance. So what we want to do is to profile the gene expression of these dance or speech region. So let me break down what this means. In a nutshell, in all our bodies, we're full of cells, and all our cells carry the same DNA. But what makes these cells be skin cells and these cells inside our brains be brain cells, or as we call them neurons, is that they have different gene expression patterns. Namely, as if they had different dimers, there are different genes that get turned up and different genes that get turned down. So in this artistic visualization of gene expression that you have here, imagine as if the orange dots represent genes that get turned up in these primary motor cortex neurons, and these same genes here in blue get turned down in these skin cells. So although we know several of these differences in gene expression in different cell types, what we definitely have no clue about is what is the gene expression that makes a speech cell be a speech cell and the dance cell be a dance cell. So this is what we will seek to find out with this great technology that's called single nucleus RNA sequencing. But of course, in order to study this gene expression in humans, we need to have post-mortem human brains that we thankfully have thanks to the generous donation of several individuals to science. But the problem is that these brains come in frozen slices, and our locations from the fMRI are in a 3D space. So we're currently reconstructing these frozen slices in a 3D space in order to be able to pinpoint to the exact brain region that does speech and dance and to profile its gene expression. And here you can see our team's first successful 3D reconstruction of a frozen human brain in collaboration with a great team from Harvard. Another plan that we have is to sequence in high quality genomes, the DNAs of individuals that really excel and do very well in the behaviors of speech and dance. For example, take opera singers or professional dancers. Are there any signatures in their DNA that can predict their abilities to speak or sing and dance in these ways compared to the average population? And what if we do the same thing in populations that show speech deficits or dance deficits, as those, for example, seen in autism spectrum disorders or in Parkinson's disease? So we really want to shed light on the full genetic architecture of speech and dance. Now, the last part, let's get to therapy. Because interestingly, one of these two behaviors, dance, is already being used as a therapeutic practice that has been shown to alleviate significantly several body motor deficits, for example, in Parkinson's disease. So in Parkinson's disease, we observe tremor, bradykinesia, slow movements, or gait freezing, and dance has been shown to alleviate these aspects. So our question after having put all this picture of evolution and development together is, what if we also test if dance, this motor practice, can also alleviate another motor deficit in Parkinson's disease, namely speech? Because there are definitely speech deficits in Parkinson's disease, for example, deficits in pitch, intensity, or speech rate, and they are present in a very high percentage of the population of Parkinson's disease. And what several studies have shown is that what actually might underlie these speech deficits are specific issues in the laryngeal muscle movements and in the way these patients can or cannot close their vocal folds. So if you remember at the beginning, in one of my first slides, I showed how, for me, in order to produce this prolonged ee sound, I had to bring my vocal folds really close to each other. So these patients here suffer from this insufficient vocal fold closure, and they cannot really bring their vocal folds together, and this gives rise to several of these speech deficits. So what we want to do is to test whether by dancing, we can get improvement in these patients' speech deficits. Because our idea is that if speech and dance are so linked, so much linked in development, in evolution, even possibly in the brain areas that they employ when we are producing these behaviors, then it could be that by activating the dance brain pathway, we can stimulate activity in the speech brain pathway. And we are preparing this study in collaboration with a great team that's called Dance for Parkinson's Disease from the Mark Morris group. And they have already shown great results of how dance can be so alleviating in several motor deficits, except speech, that we are now testing together. And so far, there is one clue, one scientific clue, that is telling us that we're in the right track with this therapeutic hypothesis. And the clue comes from a study in which we were not initially involved. So this was done from a group at York University, where they had a group of Parkinson's disease individuals undergo an extensive 8-month training of weekly dance practice. So their primary outcome, what they were testing, was not speech. They wanted to find out whether dance would do anything in their depression symptoms. So first off, they found that dance was great for depression. And what they also did was that they measured their brain activity through fMRI in 4 different time points of the experiment. And when they learned about our plan to work on what dance could do in the speech deficits of these populations, they told us, why aren't you using our data to see whether we found any differential activity, any differences in the activity signals of these specific brain regions, like the laryngeal motor cortex, that we know are just associated with speech by and large and not with any dancing. And so we did this reanalysis. So this is not included in this paper. This is our reanalysis here. And we found that one of the regions that show these significant differential activity patterns during 8 months of dance training, not any speech training, is actually this dorsolaryngel motor cortex region that I've been showing all over. So what we want to study here is whether these different activation patterns that we're finding actually can be translated to real behavioral outcomes. Is it that by dancing, these patients will actually get better at speaking? So just to conclude here, I hope to have given you a good overview of how we're planning to just go through different levels of biological analysis to understand better in humans now what could be linking speech and dance. So at the level of the brain pathways, we now have the tools to address this question. We have optimized the technology for electroencephalography, and we're creating choreographies that can be done in an fMRI. And we are going to use this data in order to find out which is the gene expression profile in these brain regions, as well as sequence the genomes of humans that are really excellent in speaking, singing, or dancing, as well as in individuals that have deficits in these behaviors. And definitely there is not a better place than Rockefeller to do genome sequencing, I can really tell you that. And as of therapy, we're trying dance and what dance can do in the speech deficits of several populations that show both motor and speech deficits. And hopefully in some years, I can come back and tell you that by dancing, actually, Parkinson's disease or autism spectrum disorders patients can get better at speaking. So as Erich also said, these projects really speak to our hearts. I've been a trained and awarded dancer for several years since I can remember myself, and I have seen firsthand the health benefits that dance can have in several aspects of our health, and I really think that it remains to be proven with rigorous scientific research. Of course, all this would have been impossible without the contribution of mentors, mentees, collaborators all around the world, and of course, human and non-human participants. And of course, it would have been impossible without the necessary funding. Thank you so much for being such an engaging audience. Thank you.
They want to see a dance. Well, I'm going to just say a few wrap-up words, and then we'll take questions that Rick will lead. And just to say that I hope you'll see that the combination of the talk that we gave here really shows what we can do at a place here like Rockefeller, where we can go all the way from a complex behavior with an evolutionary perspective to the underlying brain circuits, cell types, to the genes, and even thinking about deficits and disorders. I don't know anywhere else in the world we can bring all of those things together in a rigorous manner. I know we brought it to you in a simple way, but there's a lot of detail that we're not showing you in a rigorous manner than a place like Rockefeller. And I say that having been other places. So I just want to thank your attention here and hope that this is a good example for those who are interested in knowing about what we do further, to do some crazy experiments for some interesting ideas to help understanding the human condition. Thank you.